Hello there. Welcome to Fundamentals of Biology, Lesson 12, where we're going to be looking at meiosis. So this follows on nicely from the previous session where we looked at mitosis. So if you remember, mitosis is the division of our somatic cells, so our body cells, whereas meiosis is the production of gametes. So we can see in the picture on the slide here that we can see our gametes. So we've got a sperm on the left-hand side, and on the right-hand side we have an egg, or to give it its correct term, an ovum. So what we're going to do in this session is have a look at how these are produced. So the objectives for this session then are to describe the stages of meiosis that we see in sexually reproducing organisms. Now, thankfully, if you got on okay with mitosis in the last session, what we'll find in this session is that the stages are very, very similar. So we still have prophase, metaphase, anaphase, telophase, cytokinesis, and all preceded by interphase. So a lot of the terminology will be the same as what we saw in mitosis, but there are some important differences as we'll look at as we go through. We'll also highlight some really important differences between mitosis and meiosis. So we'll explain how meiosis reduces the number of chromosomes in our gametes. So if you remember, our somatic cells are diploid, so they have two complete sets of chromosomes. One set we got from our mum, one set we got from our dad. Well, what meiosis does is it reduces the number of chromosomes in our gametes from diploid to haploid. So that means they only have one complete set of chromosomes. So we'll investigate that as well. And then we'll also talk about genetic variation and how meiosis introduces genetic variation into our gametes, and also why it's important. So that's the aim for this session then. So it's worth just going through a quick comparison between mitosis and meiosis. So if you remember from the last session, mitosis is the division of somatic cells, and we use it for development, growth, and repair. And mitosis produces two daughter cells that are genetically identical to each other. And during the process of mitosis, we conserve the number of our chromosomes. So what that means is the parent cell is diploid, so having two complete sets of chromosomes. And because we go through DNA replication during the S stage of interphase, when that parent cell divides into two, well, we get two daughter cells that are also diploid. So it conserves the number of chromosomes. If we compare that to meiosis, then what we see is that meiosis is the production of gametes. So it's not the division of gametes, it's the production of gametes. Unlike mitosis, which just undergoes one division, we see that meiosis actually undergoes two divisions. So we go through meiosis I, and then we go through a second division called Meiosis II. So we'll go Prophase II, Metaphase II, Anaphase II, Telophase II, and then cytokinesis. So at the end of meiosis, because we go through two separate divisions, we actually end up with four daughter cells. And each one of those four daughter cells are genetically different from each other. So as we'll see as we go through, we actually mix up some of our genetic material. The chromosomes there get jumbled up a little bit. And there are three specific places within meiosis, three stages where genetic variation is introduced. And I will highlight these as we go through. And what we also see, as I've mentioned previously, is that during meiosis, the chromosome number is halved. Unlike mitosis that conserves the number of chromosomes, so you get a diploid parent cell and two diploid daughter cells. In meiosis, we have a diploid parent cell, and then we end up with four haploid daughter cells. So each of our gametes, whether it's an ovum or sperm, will have just one complete set of 23 chromosomes. And on the next slide, we'll talk a little bit about why that's so important. Okay, so let's just focus on those important points then. To begin meiosis, we go through interphase, just like we saw at the beginning of mitosis. So We start off meiosis with these cells called germline cells, and they will go through interphase, so G1, and then S. So during the S stage of interphase, the DNA, the chromosomes, will be replicated. So at the end of that, each chromosome will consist of 2 sister chromatids. And then we'll go through G2, where we get replication of the centrosome. And then the cells can enter into meiosis proper. So they will enter into meiosis I. It's an important point there that before meiosis begins, our germline cells will go through interphase, including DNA replication. But there's these 2 main points that I mentioned previously. So the first one is that meiosis reduces the chromosome number from diploid to haploid. So why is that so important? Why have I already mentioned that a few times? Well, it comes down to sex. So if you think about it, our somatic cells, to function properly, need 2 complete sets of chromosomes. So they need to be diploid. 1 set of 23 from the male, 1 set of 23 from the female. Now, if you were to have diploid gametes, so diploid sperm and diploid ova, when you get fertilization, so the sperm fertilizes the ovum, that fertilized ovum or egg would end up then with 4 complete sets of chromosomes, because you'd be getting 2 from the sperm and 2 from the ovum. So now you would end up with a fertilized egg that, instead of having 46 chromosomes in total, would have 92. And if you imagine that that individual grew up and then reproduced, well, their offspring would have 184 chromosomes, and you would keep doubling with each subsequent generation, which would be unsustainable, because you just end up with far too many chromosomes inside the nuclei, and the cell wouldn't be able to function. So what we do when we produce our gametes, so when we produce our eggs and sperm, is that we reduce the number of chromosomes by half, so that each sperm will have 23 chromosomes, each ovum will have 23 chromosomes. So when fertilization occurs, we end up with a fertilized ovum with 46 chromosomes. So that's the first really important point about meiosis. It reduces the number of chromosomes from diploid, meaning 2 sets, to haploid, which is just 1 set of chromosomes. The next really important point about meiosis is that it jumbles up the chromosomes within the cells during the process of meiosis. So it introduces genetic variation. Each of the sperm that are produced and each of the ova that are produced will have different genetic makeups. And this is incredibly important for something like evolution through natural selection. So what you are essentially trying to do is produce offspring that have different genetic makeups, in the hope that you'll be producing offspring that are fit enough to survive in the environment, make it to maturity, and then reproduce themselves. The world is constantly changing and things are constantly evolving. So you might be familiar with something like the evolutionary arms race that exists between predator and prey. So this would be where a predator evolves to be slightly faster or evolves the ability to make tighter turns at high speed. And then the prey animal will evolve ways to counteract that. So they will evolve to be stronger or faster, or they will evolve to be able to make even tighter turns, or they may evolve to be camouflaged, or they will evolve some way of countering the predator. The predator will evolve to overcome that. So you get this constant shifting forward of this relationship between predator and prey. But you also get a similar relationship between parasites and hosts. Bacteria and viruses are constantly evolving, and we have to constantly evolve to try and counter them. It is through this jumbling up of our genetic material during meiosis that you would hope to produce some offspring that have the right genetic makeup to enable them to survive within the ever-changing environment. So meiosis is really, really important for this introduction of genetic variation, so our offspring can survive or have a good chance of surviving in the ever-changing world. So let's do a quick run through of the basic stages of meiosis, and then we'll focus in a little bit more detail. So what we can see on the diagram on the right hand side here is a main breakdown of interphase and then also Meiosis I and Meiosis II. So if we start at the top of the diagram here, we can see that we're in interphase. And what we can see here, so this is a diploid cell, and we're going through interphase. So we're entering into the S stage of interphase where the genetic material or the DNA replicates. So we have a homologous pair of chromosomes in a diploid parent cell. The chromosomes replicate. So now we have homologous chromosomes, and each chromosome consists of 2 sister chromatids. Okay, so within sexually reproducing animals, what we would have now are a type of cell. So in males, it would be a type of cell called a primary spermatocyte. And I'll show you these on a diagram in a bit. So we'd have a primary spermatocyte, and then we would have in females a primary oocyte. And these are now ready to undergo meiosis. So the first thing that happens, we're not looking at genetic variation here at the moment, we're just looking at chromosome reduction. So the first thing that happens in Meiosis I is that our homologous chromosomes separate. So what Meiosis I does is it takes our diploid parent cell and it produces 2 haploid daughter cells. Okay, so if we look at what's happening here, we've got one of the homologous pairs of chromosomes will go off into this daughter cell, and then the other homologue will go off into this daughter cell. So in humans, we could say that this parent cell here would have 46 chromosomes. Each chromosome consists of 2 sister chromatids. So we've got 46. And then at the end of Meiosis I, we have 2 daughter cells. Each one has 23 chromosomes, so they are haploid. Each chromosome, though, still consists of 2 sister chromatids. Okay, so that's what we get at the end of Meiosis I. We end up with 2 haploid daughter cells. We then go through Meiosis II, and we can see Meiosis II here, and what that does is it separates the sister chromatids. So we can see if we follow this daughter cell, we can see the chromosome here. So in a human, there would be 23 of these in this daughter cell, and we can see one sister chromatid will go off into this daughter cell, the other one will go off into this daughter cell. And then we can see the same thing over here. So what we would end up with at the end of Meiosis II are 4 daughter cells that are each haploid. So in humans, each one of these 4 daughter cells will have 23 chromosomes. So hopefully that makes sense as we just do the quick run through of how meiosis reduces the number of chromosomes. We start off with a diploid parent cell. We undergo meiosis I that separates the homologous chromosomes. So we end up at the end of meiosis I with 2 daughter cells that are now haploid. But each chromosome, because it replicated up here during interphase, each chromosome consists of 2 sister chromatids. And what Meiosis II does is it separates those sister chromatids. So we end up at the end of Meiosis II with 4 daughter cells that are each haploid. So each one of these 4 cells will have 23 chromosomes. And we can see that in the text here. So in the first cell division, so Meiosis I, homologous chromosomes separate. Meiosis I results in 2 haploid daughter cells, but the chromosomes are still replicated in the sense that they still have 2 sister chromatids. In Meiosis II, those sister chromatids separate. So Meiosis II results in those 4 daughter haploid cells that have unreplicated chromosomes. So they are just individual chromatids, if you like, but we would refer to them as single chromosomes. Okay, so hopefully that makes sense in terms of how meiosis reduces the chromosome number from diploid to haploid. That important step occurs during meiosis I, where we separate our homologous pairs. So next we want to have a look at where the genetic variation is introduced. So what we can see on the diagram here are the stages of meiosis I. So before meiosis I begins, remember, we have the parent cell that undergoes interphase. So we go through G1, S, and G2. and then that cell is able to enter into meiosis I. And we go through the same stages, or the same basic stages that we saw with mitosis in the previous session. So we start off with prophase I, and then we move into metaphase I, then anaphase I and telophase I, followed by cytokinesis. And the general basic mechanics behind each stage is very, very similar, in terms that we have the formation of a mitotic spindle, we have the breaking apart of the nuclear envelope, we have the aligning of the chromosomes along the metaphase plate, and then the separating of the chromosomes. So all of those things are very, very similar. But there are some key differences, particularly in meiosis I. So let's go through this. On the next slide, I've got some text that so you can make some notes. So let's just quickly go through each of these stages, and I'll talk to you a little bit about how and when the genetic variation is introduced. So let's start off with prophase I then. This is probably the most important part of meiosis, and it actually takes up the vast majority of the time of meiosis. So about 90% of the time a cell spends in meiosis is spent in prophase I. A couple of really important things happen here. We've got the basic stuff, so you can see up here we've got our two centrosomes, we've got the formation of the mitotic spindle through those microtubules, we've got the breaking apart of the nuclear envelope. We kind of combine pro-metaphase in here as well, so we just go from prophase to metaphase. But the important bits happen with the actual chromosomes themselves. So what you can see here in this diagram is that our homologous pairs of chromosomes, so you can see here you've got the red and the blues that represent the homologous pairs of chromosomes. Well, during prophase I, they will actually line up alongside each other, and they align next to each other, so the same genes are next to each other, and then they form something called a tetrad. So this tetrad, you can see three tetrads here. So this is a tetrad here where you've got a homologous pair of chromosomes aligned next to each other, another one down here, and another one here. These homologous pairs are now attached to each other, and what they will then do is something called genetic recombination, or simply put, crossing over. So what they will actually do is the non-sister chromatids of the homologous pair will actually cross over each other and swap a bit of their genetic material. So the chromosomes from the homologous pair will actually swap some of the DNA with each other. This introduces genetic variation. You could imagine if we talk about chromosome number 1, so the chromosome number 1 we got from mum and the one we got from dad will align next to each other, and then the chromosomes will then cross over each other, and a bit of mum's one will be snipped off and it will swap over with the same bit from dad's. So what you end up with at the end of prophase I are chromosomes that have now been jumbled up. They no longer have the same genetic makeup that they had before prophase I began. Prophase I, we get the lining up of the homologous chromosomes, so they undergo something called synapsis, which is where they line up and then cross over each other, exchanging a bit of genetic material. Okay, so that's prophase I. What happens next is metaphase I. We can see here now we've got a fully formed mitotic spindle, just as we saw in the last session with mitosis, microtubules attached to the chromosomes at the kinetochores, which are located at the centromeres. So all those words again that I used in the last session. And then the microtubules then are able to shift the chromosomes around so that they are aligned along the metaphase plate. But there's a really important difference here between what we saw in mitosis and what we see here. If you look at this diagram, you'll see that our homologous pairs of chromosomes are still attached to each other. They're still aligned in that tetrad. We can see a homologous pair here, another pair here, and another pair here. Now, obviously, this is just showing 6 chromosomes, but if we were looking at a human cell here, we would have 46 chromosomes, so there would be 23 of these tetrads all aligned along the metaphase plate. Now, metaphase I is another stage in which we get genetic variation, and that is because the way that these chromosomes align along the metaphase plate is random. And what I mean by that is, let's say the red chromosomes here we got from dad and the blue we got from mum. Okay, so red we got from dad, blue we got from mum. If you look at this diagram, you can see that the way that these are lined up now, so 2 of the red ones from dad are going to go up into this daughter cell. So these are going to divide in a minute, and they're going to go up here. And then we've got this blue one from mum that is going to go up here. And that means that the daughter cell that comes from this one will have these 2 blue ones from mum and this 1 red one from dad. So they're going to move down here. Now, the way that these arrange like this are completely random. So you could have a situation where you've got all 3 red ones from dad going up into this daughter cell and all 3 blue ones from mum coming down here. Or it might be a red, blue, red going up here. Or it might be all 3 blues going up here. So this arrangement of, or this random alignment of chromosomes along the metaphase plate during metaphase I introduces another stage of genetic variation, because the alignment of the chromosomes is completely random. So hopefully that one makes sense as well. So we've been through prophase I, we've now gone through metaphase I. We can then move into anaphase I, which is where our homologous chromosomes are separated. Just like we saw in anaphase with mitosis, where the sister chromatids were pulled apart, in anaphase I of meiosis I, it's the homologous chromosomes that are pulled apart. So if we look closer at this diagram, you can see in metaphase I, so let's just take this first tetrad on the left-hand side here. So the red chromosome was from dad, blue from mum. If we look over here at what's happening in anaphase, they are being separated. But you can still see that each chromosome consists of 2 sister chromatids. And what you can also see when you look at the chromosomes here in this diagram of anaphase I, that the chromosomes are different. You've got little red patches on this blue one over here. You've got blue patches on this red one here. And it's different all the way through. Why is that? Well, we go back to prophase I, where crossing over occurred. So the genetic material, the chromosomes, jumbled up. They swapped some of their DNA. And we can see that with this color configuration in this diagram here. So you've got genetic variation there through crossing over in prophase I and the random alignment of the homologs during metaphase I. At the end of anaphase I, we then move into telophase I, where just like in mitosis, we start to get the reformation of the nuclear envelope and cytokinesis, which pinches the cell apart. So we finish meiosis I with 2 daughter cells that are now haploid. And they are haploid because in anaphase I, we separated the homologous chromosomes. Let's just put some text to that then so you can make some notes. Prophase I will typically occupy more than 90% of the time spent in meiosis. During prophase I, we get synapsis. And this is where homologous pairs loosely pair up and they align themselves gene by gene into what we call tetrads. So a tetrad will be that pairs of homologous chromosomes aligned next to each other. Once that happens, they will start to cross over. So we get this genetic recombination where they swap a bit of genetic material, or they'll swap a bit of DNA. So this is where we get the introduction of genetic variation. We can then move into metaphase I. This is where our tetrads, so our homologous pairs of chromosomes, line up randomly at the metaphase plate. So one of the chromosomes, one of the homologous pair facing each pole, that will eventually become our daughter cells. So again, because this alignment is random, it also introduces genetic variation. And then we just go through anaphase I, which will then separate the homologous pairs. So that's what's turning our daughter cells into haploid cells, because we're separating our cells out. So we go from a 46 chromosome parent cell to 2 23 chromosome daughter cells in humans. And there we go. Meiosis I produces two haploid daughter cells that are genetically different. Why are they genetically different? Well, because of Prophase I and Metaphase I. Okay, so that's a run through of Meiosis I. And now that will lead us in to Meiosis II. So Meiosis II is a lot more straightforward than Meiosis I, thankfully. And it's very, very similar to what we see in mitosis, because essentially what we're doing is we're just separating our sister chromatids. So let's go through the stages. Just like previously, I've got some text on the next slide so you can make some notes. An important point to make is that the daughter cells that we got at the end of Meiosis I, they do not go through any further genetic replication between Meiosis I and Meiosis II. So we start Meiosis II in Prophase II, and we can see our chromosomes here. So you can see that you've got the genetic variation that we got from Prophase I and Metaphase I here. So we've got a nice jumbled up mix of chromosomes. And then in Prophase I, we'll just get in Prophase II, we'll just get normal prophase things going on. So we'll get the formation of the mitotic spindle, the breaking apart of the nuclear envelope, the condensing of the chromosomes, just like we've seen in prophase before. And then we move into Metaphase II. Now, Metaphase II is the third place or the third stage where we get genetic variation introduced. And that is because the way that the chromosomes align along the metaphase plate is random again. If we see here, because the sister chromatids of each chromosome are now different because of what happened in Prophase I, because of crossing over, each of the sister chromatids of our chromosomes are no longer identical. So if we take Metaphase II and we'll look at this daughter cell up here, we look at chromosome here, the bottom of this daughter cell, we can see that the sister chromatid on the right is completely blue, yet the sister chromatid on the left has these two red ends to it. And that's because during Prophase I, it swapped those bits of genetic material with this sister chromatid down here, or this non-sister chromatid of the homologue, which is a bit of a mouthful. But you can see how these were swapped with this one up here during Prophase I. So these two sister chromatids are no longer identical. So depending on how these are arranged during Metaphase II, so it could arrange like this with the blue sister chromatid, which is going to move off into this daughter cell, and this sister chromatid with the red ends is going to move into this daughter cell. Well, they could just as easily have been arranged in the opposite direction. So the 100% blue one would move into this daughter cell, and the one with the red ends would move into this daughter cell. And it's the same with the rest of them. So obviously keep in mind, we're only looking at, in this case, 3 chromosomes in here, but during human meiosis, there would be 23 of these all aligned along the metaphase plate. You'll have an awful lot of different possible variations in which direction the sister chromatids are going to go. During Metaphase II, we get the random alignment of the chromosomes along the metaphase plate. So that also introduces genetic variation. We then move into Anaphase II, which, just like we saw in anaphase of mitosis, will separate the sister chromatids. So one set of sister chromatids will move into the daughter cell that is going to come from here, or from this right side of the cell. The other set will go to the left over here, and the same for this cell down here. And then we move from Anaphase II into Telophase II. So we just get the decondensing of the chromosomes, reformation of the nuclear envelope, and then cytokinesis will also begin there as well, which you can see with the cleavage furrow. Each of these two daughter cells that we produced at the end of Meiosis I are now separating into 2 daughter cells of their own. So we end up at the end of Meiosis II with 4 daughter cells. So you can see 2 up here and then 2 more down here. So we finish Meiosis II with 4 daughter cells that are genetically different from each other, and they are also haploid. So let's go through the key points for Meiosis II then. So Meiosis II is very similar to mitosis, as it separates the sister chromatids. In Metaphase II, the sister chromatids are arranged randomly at the metaphase plate. So again, this is our third place or third stage where we get the introduction of genetic variation. Because of crossing over in Meiosis I during Prophase I, the two sister chromatids of each chromosome are no longer genetically identical. So depending on which daughter cell they move into, well, they're going to be able to influence that particular daughter cell. And then at the end of Meiosis II, we have 4 genetically distinct daughter cells, each with a haploid set of chromosomes. So that's meiosis, essentially. We go through 2 different divisions, so Meiosis I and Meiosis II. We get a reduction in the number of chromosomes, so we go from diploid to haploid, and we also introduce genetic variation in Prophase I, Metaphase I, and Anaphase I. So what I want to show you next is the animation from the NDSU virtual cell again, and this will run through the stages of meiosis. So look out for those key points, where you get genetic variation, and then also where we get the reduction of chromosome number. And just like in the animation for mitosis, she will move through it quite quickly to begin with, and then go through each stage more slowly, and then recap again at the end. Have a look through this, and then we will have a catch up afterwards.
Many organisms pass their genes to their offspring through sexual reproduction. This begins when 2 gametes unite to form an embryo that is genetically unique from the parent organisms. The embryo then grows into an adult who in turn passes their genetic information on to their own offspring. Gametes are formed through a process called meiosis. The cells that undergo meiosis to produce the gametes are called germline cells. In diploid organisms, germline cells have 2 copies of each chromosome. Germline cells undergo meiosis to produce haploid gametes, which only have 1 copy of each chromosome. These haploid gametes fuse to form a diploid embryo that grows into the adult. Meiosis is just one step in the life cycle of a germline cell. Similar to mitosis, the cells also pass through the interphase, G1, S, and G2 stages before they enter meiosis. The DNA inside a germline cell is duplicated before meiosis begins during the S phase. The duplicated germline chromosomes are called sister chromatids. These chromatids remain attached to each other until the second cell division event in meiosis. There are 2 cell division events during meiosis. The first division, Meiosis I, results in 2 unique daughter cells that have half the amount of DNA as the parent germline cell. The second division, Meiosis II, results in 4 unique haploid cells that only have 1 copy of each chromosome. These haploid cells are the gametes that could go on to produce an offspring through sexual reproduction. Let's look more closely at each of the division events. Meiosis begins with Prophase I. In this stage, the DNA condenses to form chromosomes. Here we see the duplicated sister chromatids joined together at the centromere. They stay fused at the centromere throughout Meiosis I. Next, each pair of homologous chromosomes undergoes synapsis to form a complex involving 2 pairs of sister chromatids. Chromosomal material is exchanged between the 2 pairs of sister chromatids. This event is called recombination, or more commonly, crossing over. After crossing over, the sister chromatids for each chromosome are no longer identical to one another. This is one of the reasons why no 2 siblings, aside from twins, are genetically identical. There are several more key steps in Prophase I. The nuclear membrane begins to break down. Then, the 2 centrosomes migrate to opposite ends of the cell and microtubules appear. The microtubules then attach to the chromosomes. The next phase of Meiosis I is called Metaphase I. Here, the synapsed chromosomes align at the equator of the cell. The chromosomes align randomly, which results in different combinations each time meiosis occurs. The next phase is Anaphase I. During this phase, homologous chromosomes separate and migrate to the 2 poles of the cell. Importantly, the sister chromatids remain attached at their centromeres. The final steps of meiosis I are telophase I and cytokinesis. Here, the cell divides into two daughter cells. Each of these two cells now undergo meiosis II. Meiosis II is similar to mitosis. The first stage of meiosis II is prophase II. Again, chromosomes condense, the nuclear envelope breaks down, and the spindle apparatus forms. The major difference between prophase II and prophase I is the fact that the daughter cells have only one copy of each homologous chromosome. So in prophase II, there is no synapsis of homologous chromosomes or crossing over. In metaphase II, the chromosomes align at the equator of the cell. Again, the alignment is random. Since the sister chromatids are no longer identical, there will be many different possible ways for these chromosomes to align. In Anaphase II, the sister chromatids are pulled apart as the microtubules shorten. Also, the ends of the cell are pushed farther apart as microtubules elongate. In telophase II, the nuclear membrane reforms and the cytoplasm is divided into the two haploid daughter cells. This division is called cytokinesis. Since meiosis II began with two cells, and each of those cells were split into two cells, we now have four unique haploid cells at the end of meiosis. These cells are gametes. Two gametes, one from a father and one from a mother, may fuse to produce a diploid embryo. The resulting embryo then grows through many cycles of mitosis.
Okay, so let's, as always, finish off with a quick recap quiz to see how you've gotten on. So I'll read through the questions, give you some time to answer them, and then we'll go through the answers together. So question number 1, how many daughter cells does meiosis produce? Question number 2, what is meiosis used for? So what does it make? Number 3, how many chromosomes do human gametes have? Number 4, which 3 stages of meiosis introduce genetic variation? And then number 5, why is the reduction of chromosome number necessary? Okay, so have a go at answering those 5 questions. I'll give you a few minutes to answer them, and then we'll go through the answers together. Okay, so welcome back. Hopefully you've had a chance to answer those. So let's go through the questions, or go through the answers together. So number 1, how many daughter cells does meiosis produce? Well, the answer is 4. So we get 2 from meiosis I, and then they divide further, so we end up with 4. What is meiosis used for? So question number 2. Well, the answer is the production of gametes. So it makes gametes, sperm, and ova. Number 3, how many chromosomes do human gametes have? So hopefully you all got this right. 23. So they are haploid, as opposed to the diploid somatic cells. And number 4, which 3 stages of meiosis introduce genetic variation? Well, they are prophase I, metaphase I, and metaphase II. And then number 5, why is the reduction of chromosome number necessary? And the answer is that haploid gametes enable the fertilized ovum to be diploid. Okay, so we reduce our number of chromosomes by half, which means that our fertilized ovum or our fertilized egg will then be diploid. And then it can undergo mitosis to produce an actual human in our case. Okay, so hopefully you did okay with those, but as with everything else, you might need to go over this a few times, go over the key points, and that might help it to sort of sink in a bit. Okay, so that brings us to the end of this session then. Hopefully now you would be able to describe the main stages of meiosis in sexually reproducing organisms. You should be able to explain how meiosis reduces the number of chromosomes from diploid to haploid, and not just how, but also why. And then also explain how and why meiosis introduces genetic variation. Anyway, that brings us to the end of this session. Hopefully it was useful. Thank you very much. I'll see you in the next session.